We will look at 2D HSQC Toxi experiment today. In the last class we looked at protein structure analysis using HSQC, but there is a very next important experiment which is used often in NMR of proteins is 2D HSQC and 3D HSQC Toxi. So, let us have a look at how this is designed and developed. In fact, this will be very useful for us to understand 3D NMR in the next part when we start. So, 2D HSQC spectroscopy basically is the same as combining an HSQC and Toxi in a same pulse sequence. So, let me now take you through a very simple approach how these two experiments, HSQC is one experiment, Toxi is another, how these two can be actually combined and we can generate 2D HSQC Toxi, which will give you a very good rich information compared to HSQC or Toxi separately. So, this is what is shown here, that the essential idea is we are trying to combine a simple HSQC experiment which we have already looked at in detail with a proton-proton Toxi experiment, which also we have looked in the homonuclear part. So, now we are combining an heteronuclear NMR experiment with a homonuclear NMR experiment, and both were 2D experiments as we saw in the previous lectures, but now we are combining them to create a new 2D experiment. So, this is also going to be a 2D experiment which will be called as HSQC Toxi. So, this as I said will form a basis for going into 3D NMR, which we will see how 3D NMR are built. So, it will be similar to what I show you here, the 3D NMR experiments are built. So, let us start again from a basics or recap of HSQC pulse sequence. So, this is what we saw in the HSQC part. You start from a proton excitation, you excite the proton pulse, then there is what is this called as inept experiment, inept module. So, in a inept module you transfer the magnetization from proton to carbon, or it can be proton to nitrogen as well in the case of nitrogen HSQC, and this is done through this series of these two 180s here, and they are exactly in the center of delays, two delays. So, there is this long delay which is 1 by 4j, another delay 1 by 4j on either side. So, this j is according to what we saw is depending on the coupling value. So, if j is 140 hertz, typically the value we get is 1.8 milliseconds here and 1.8 here. Now, after this pulse excitation is over, we take it back to z axis, the proton pulse, and carbon is excited now to bring it to the xy plane, and during this period the carbon is in the xy plane and it evolves according to its chemical shift value during this period, and that is during this T1 half, T1 half, but if you combine these two it becomes T1. So, again repeating why have we split this T1 into two parts, because we want in the middle we want to have a 180 degree pulse on proton, and this helps to decouple the carbon from proton. So, any 180 at the center of evolution, not on the nucleus, on another nucleus, helps to decouple that nucleus from this evolving nuclei. So, this is the principle behind this idea, that if carbon is evolving and I put a 180 degree in the center, it decouples my carbon from proton. So, there is no proton carbon coupling which is present during this period, and following this period we then transfer the polarization back to proton, and this is during the reverse inept. So, in the reverse inept we basically go back to the proton magnetization or polarization, which is something which we have seen in HSQC. This is again the same as the inept here. So, we have 1 by 4j, 1 by 4j, and here also we have 1 by 4j and 1 by 4j. So, you can see that this part is same as this part, there is no difference. It is just that this is called as forward inept and this is called as a reverse inept. Now, we can see here the magnetization at this point will be completely on the hydrogen proton, and then what we did in the HSQC was we started evolving this magnetization, we started detecting this magnetization, and during that period we had the decoupling on the carbon present. But in HSQC Toxi, now we want to combine this HSQC with a Toxi. So, what I need to do now, I need to add a Toxi part in this here, and then that becomes an HSQC and a Toxi. So, before we look at the Toxi part here, just look at the magnetization again, how the magnetization is going from proton to carbon. So, now look at this schematic drawing here of a molecule. So, let us say your molecular structure is something like this. So, what we are looking at here in the first part here is that we started from a proton which is we can label as I, and I transfer the polarization to carbon through this inept. Okay. And then in HSQC that is what is shown here, we transfer to carbon and we had this carbon chemical shift evolution, and then we transferred it back to proton, and this is exactly what, this is basically what we did in HSQC. But now what we will do is we will add an additional part, and that is the TOCSY part. So, you can see here this is the TOCSY mixing now. So, what is the part doing here? It is basically mixing or transferring the magnetization from one proton to another proton. So, if you look at this point here where the arrow is pointing, we are basically on the proton magnetization. Okay. So, we are in the I, that is HI, which is this HI. I have taken this HI as an arbitrary HI. So, now from here onwards it has, now it is in the xy plane, we have not detecting this proton yet, it is in the xy plane. Now, if I apply a mixing like this for some duration, if you recollect, the duration time for this should be around 60 to 80 milliseconds long. So, during this period the magnetization from one proton or that I proton is transferred to every other proton in the spin system. So, remember, we have to remember that the magnetization goes within the spin system only, it does not go out of the spin system. The transfer of magnetization from one proton to the remaining proton takes place only within a given network of coupled spins. So, we are having all J coupling to each other. So, this hydrogen is J coupled to this hydrogen is J coupled to this hydrogen, but this need not be directly coupled to here. So, I is coupled to J, J is coupled to K, and therefore, I gets coupled to K through J, and that is the long range interaction which is through TOCSY mixing, happens through TOCSY mixing. Now, during this period we use the power level etc. similar to what we do in a regular TOCSY. So, this is nothing different from a regular 2D TOCSY, but you can see now what I have done here, I have taken an HSQC part and simply added a TOCSY mixing. So, this is how we design or we construct multi-dimensional NMR experiments. We take one experiment and we concatenate, meaning we add to it or append another experiment, and the combination is done in a way such that the required information is given to comes out. So, we will look at the information now. So, now during this period T2, the magnetization evolves on that all the hydrogens wherever the magnetization has been transferred, because now this was in xy plane at this position here, from here it got transferred to here, and there it's there these protons are also now in the xy plane, and all of them now start getting evolved or they start getting detected during the T2 period, and during that period we are decoupling as usual. Now, this chemical shifts of each of this hydrogen is just written as H1, HI, Hj, Hk. So, they will evolve during T2, and we can say that their evolution is captured as cosine omega Hi, Hj and Hk T2. So, now if you look at these correlations, what we have done with this experiment now, we have correlated or connected the chemical shift of carbon of one carbon of I to another to its own proton, which is HSQC. So, this combination, that is HCI T1 and HI T2 is nothing but the HSQC peak, which all of us have now seen it in the previous lectures. This is what we got in the HSQC. But what additional information now we have obtained because of the TOCSY is that we have correlating now Ci with also Hj. So, you see this is not a direct coupling, because that was 2 bonds away. So, you can see that J is here, this carbon Ci is actually 2 bonds away. So, I remember I told you in the HSQC we do not tune the coupling to 2 bond, we tune the coupling only to 1 bond, carbon and proton. But here you can see now we have got information of Ci chemical shift correlated with Hj. Not only that, we have now got Ci chemical shift correlated to Hk as well, because all this combination will come, this cosine into this cosine, this cosine into this cosine, and this cosine into this cosine. So, these 3 combinations, all of them will come, which I will show you shortly. So, these 3 have been correlated with this carbon, which would not have happened in a normal HSQC. In a normal HSQC, I would not have seen the interaction or correlation of this chemical shift of this carbon with this proton and this proton because of this delay which is tuned only to this coupling. So, this is mathematically is what is shown here. You can see that the FID which I finally detect here, this T2 is looks like this now. You have cosine C1 into T1, cosine C1 T1 omega, which is this, into the H1 T2, which is this. So, this is colored in red because it is now the same as an HSQC peak. This is no different from HSQC. If you go back in slides in the previous lecture, you will notice that we did see this correlation also in HSQC. So, there is no different from that, but additionally we have got two more correlation for this particular molecule. If the molecule is longer, we would have got even more longer correlations, long range correlations. What we are seeing now right now in this case, in this schematic drawing, we have only Hj and Hk, but Ci is getting correlated with Hj and Ci is also correlating with Hk. So, these are the two special additional chemical shift correlations which we would not have got in a normal HSQC, but by adding this TOCSY part we are able to get this information. Okay. So, this is what is shown here, an additional correlation, that is all the same, that is now you can see this. Suppose if I do the detection on this particular proton, suppose I do, transfer the magnetization from Hj to Cj. Okay. So, suppose I now if I look at Hj into Cj interactions, this is again direct HSQC peak, but now because of the TOCSY, Hj will also correlate with Hi and Hj will also correlate with Hk. Okay. So, now when we are looking at this combination, so let me write this here. So, now suppose we focus on this pair, this hydrogen is correlated with this hydrogen is correlated with this because of TOCSY. The TOCSY is helping us to correlate this proton to this proton. Now, this is what is shown in this particular here picture in the bottom line. The bottom here you can see. So, we are looking at this pair. So, now this is an HSQC peak, this is what we would normally get in HSQC, but in this HSQC TOCSY we will also correlate this carbon with this proton because of this transfer, and this carbon also gets correlated to this proton because of this transfer here. So, that is what is shown here. So, that is also well possible in TOCSY. So, TOCSY basically correlates this carbon with these 3 protons, this carbon with these 3 protons and this carbon with these 3 protons. So, all carbons get correlated with all protons. So, HSQC TOCSY is an experiment, a very beautiful experiment which helps us to find all connectivity between all protons and all carbons, and that is something you will not get in a regular TOCSY, neither you will get in a regular HSQC. You need this combination HSQC TOCSY to connect all carbons with all protons in a given spin system. So, you have to remember this is only in a given spin system. If there was a break here and the remaining carbons were away from the break, we cannot connect that. We need to connect, we can connect only within a family of coupled protons. So, let us look at the peak pattern, what you would expect in a regular HSQC TOCSY, and this is a simple amino acid alanine which is given here. So, you can see in alanine we have this proton CH3 and there is a proton H which is C alpha, H alpha. So, now in a regular HSQC what do you expect to get? In a regular HSQC you expect to get the correlation from this carbon to this proton, these 2 pairs, one pair, and another pair would be this pair. So, you basically get 2 peaks. Now, typically C alpha comes around C beta. So, this is a slight error here, let me correct this. This should be C alpha and this should be C beta. Okay. So, this is what we should keep in mind. So, now typically what you would expect is a C beta will correlate with H beta. Okay. So, that will come around 20 ppm and H alpha will correlate with HC alpha. So, this is HSQC part. Okay. So, this is HSQC part. So, I can write it here. This is basically the HSQC part. Okay. So, this is not different, this is nothing which is new, we already have seen this with several examples. We took glutamine, we saw for other molecules. Now, what is the additional information that we get from TOCSY? The TOCSY now gives you this part, because C beta is now correlated with H alpha, that is this green peak here. Okay. So, and C alpha is correlated with H beta and that is this peak here, because you can see the C alpha chemical shift is correlated with H beta and C beta here is correlated with H alpha. Okay. So, but please keep in mind this is not a diagonal peak. So, I should write it here clear, this is not diagonal, okay. This is already a cross peak. So, this is also a cross peak and this is also a cross peak. So, it looks like as if this looks like a diagonal for some people and this looks like cross peak, but according to this is already a cross peak, because remember this is proton axis and this is carbon axis. So, there is no question that this is a cross peak, this is actually not a diagonal peak, but now additional cross peaks are available now, and that additional cross peak is coming because of the Toxi mixing which we saw in the previous slide, that this hydrogen is now correlated with this carbon and this proton is correlated with this carbon. Okay. So, we can see this is the cross talk between two carbons. So, the entire spin system now is available based on this pattern. So, you see if I look at this square, or this is not actually a square, a rectangular pattern. So, along this line, if you look along this line, I am getting from one carbon to all the hydrogens in the molecule in a given spin system. Although I may say molecule, but a molecule can have two spin systems as we saw in the heptanone case. So, if basically within one spin system, which is in amino acids, as I mentioned last time, one amino acid is actually one family, one spin system. So, in a one amino acid you can correlate one carbon with all the hydrogens or protons in that molecule. Similarly, you can see another carbon in the same molecule, I correlate with all the hydrogen. So, basically all hydrogens with one carbon and all hydrogens again with another carbon. So, if I look at them, they will look like this, that all hydrogens will come here and all carbons will come here. Okay. So, let us see now a little more examples, another few examples of this system. So, this is the Toxi part. So, this dimension what you are seeing in the horizontal axis is proton, but it is because we are getting all the protons in this line, we are calling it as a Toxi line or a Toxi pattern, because all protons are connected to each other. So, let us say that we have these two molecules. So, for example, we have this molecule which is an amino acid here, and this is glutamic acid or aspartic acid. Now, here you can see that there is this HSQC peak here, and what is this peak here? This is the beta proton. Okay. So, this is the beta, this is alanine here. So, you can see for alanine we can see the same pattern which I showed you earlier, these two peaks, and then this is this proton, because it comes downfield shifted. Remember, we mentioned this in the previous part when we looked at HSQC, that H alpha comes somewhere around 50-55 ppm and H beta comes around 1.5 ppm, and this is something which we saw here just in the previous slide as well. Now, if you look at this particular case, now in HSQC Toxi I am able to now get additional peaks, and these additional peaks are because of this interaction. So, this peak H beta is correlated all the way to H alpha, and that is the Toxi pattern, this H alpha and H beta. Similarly, this H beta and H alpha, they are connected to each other, and that is a Toxi. So, I am seeing this Toxi peak here, these two are repeated here, and one line corresponds to the H beta, C beta here, which is 20 ppm, and another line corresponds to C alpha, which is around 55 ppm. And this is something we saw in the previous slide, and this is just a repetition of that. So, how does HSQC Toxi help us in assignment? So, remember, one of the goal in protein NMR, which we will see eventually, if we want to assign a protein. So, we are trying to assign amino acids. So, suppose I have 100 amino acid in my protein, I have 100 spin systems in the molecule. Okay. So, therefore, I need to identify each and every spin system from my spectrum. So, this experiment Toxi helps us to do that. So, let us see how it helps. Suppose I have a mixture of valine and leucine, and this is something which I told you in a protein, we will have many amino acids like this. In fact, we may have many valines, we will have many leucines. So, you will end up with a mixture of many amino acids. So, it is similar to a mixture, although they are all joined together by a peptide bond, but each amino acid is a molecule, is a separate molecule, is a separate family. So, you can imagine a protein chain which consists of many amino acids joined together actually as a mixture of molecules, mixture of amino acids. So, as far as TOCSY is concerned, they are all similar to having 10 valine, 10 leucines, 10 alanines and so on. So, let us say that a simple example here, we have a valine and a leucine mixed together. So, how will the spectra look in HSQC TOCSY and how do we identify the molecules? It is shown here. So, this is the HSQC of the mixture. Okay, so, now here you may find it difficult to say which is valine, which is leucine. You may refer to a database or a textbook, but remember in biomolecules this is never possible to say which is which. Here yes, you can say these are methyls, but methyls can also correspond to methyl from leucine or methyl from leucine here or a methyl from valine, right. So, we cannot simply say that this is methyl of valine, this is methyl of leucine, etc. These is not easy to do that. Here to some extent yes, we can guess from based on the chemical shift value which peak corresponds to leucine and which peak corresponds to valine. For example, this could be leucine based on the database and this could be valine. So, this kind of knowledge will only help here, that is the knowledge of prior structure, but knowledge of structure will not help us to say anything about this part here because they are all methyls and knowledge will not tell us about here. Okay. So, what we need to do now is to record a TOCSY. So, HSQC we will combine with TOCSY and we will record this HSQC TOCSY, which will give us this spin pattern shown here. So, here you can see that within a given molecule, that is within leucine, I will form all the connections, all the peaks will be connected to each other, whereas within valine all peaks will be connected to each other. So, by looking at the connectivity pattern like this, you can see that these two peaks which were here, they were actually connected to each other. So, these two peaks were connected to each other. So, that is why they form a square pattern if you look carefully here. Then these two peaks were connected to this peak here and this peak here, that is this peak here and a peak here. So, you can see that this family or this set of protons are actually connected to each other and they form the spin system. Okay. So, spin system is basically a network of connections between protons to each other. Now, what is the advantage? Another advantage we have is that we also get the carbon chemical shift on this axis, which earlier was not possible from a regular HSQC, from regular TOCSY. A proton-proton TOCSY, if you recall in the homonuclear part of this which we dealt in this course, we saw that there is no carbon involved. Similarly, HSQC has no TOCSY involved, but in this kind of a spectrum I am able to actually correlate carbon with proton. Not only that, I am able to get the complete spin system in the same line of protons. So, by doing this simple joining and matching by spin peak pattern, I can actually identify that all these peaks belong to one family, that is one spin system, and that could be leucine or valine depending on one information if I have any information. For example, if I know that this line belongs to leucine, then that means the entire family now becomes leucine. If I say that this could be valine based on the database, then this whole becomes valine. So, although I did not know within these two which are valine and leucine, similarly I did not know which is leucine, which is valine, but by connection of the protons with each other, now I am able to separate or distinguish leucine from valine. Now, what about the remaining spin system? So, this green color lines are basically connecting these peaks to the respective peaks in the HSQC. Now, what about the other molecule? The other molecule is here. This is not shown here. So, let me draw that for you. So, basically we are connecting this line and this will be connected to this here, connected to here, and this will be connected to this here. So, that will form another spin system which is the valine. So, you can see that the two spin system are actually very nicely separated from each other and they form a spin system. So, these two spin system therefore can be now distinguished from each other, and that is how basically the NMR experiments, NMR spectroscopy works, that we record a TOCSY like this and we try to identify the pattern like this, and from the pattern like this we try to see what kind of amino acid that could be. So, that is valine, then we assign it to valine. If it looks like a amino acid leucine or glutamine or any other amino acid, we then assign it to that respective amino acid. Similarly, we may find another spin system which are connected to each other, and they may now correspond to another amino acid, and that could be again depending on what is there in the protein. So, the protein may consist of asparagine, serine, threonine, or there may be another valine in the same molecule. So, based on that we can say there is another valine or another leucine in the molecule in the protein, but then we do not know which leucine is which. It could be leucine number 1, this could be leucine number 20 and so on. That information is not coming from here. So, you see there are two problems in protein NMR. Number 1 is given that a number of amino acids, this is something which we will repeat again in the second part, but I am just jumping ahead to tell you what is the problem that we are trying to address in protein NMR. The two challenges are, number 1, that I have, let us say, 10 leucines, 4 valine, 3 arginine and so on. So, there is multiple copies of the same amino acid. It is not that there is only one valine, not one arginine, one serine and so on. So, our task is to first figure out what are the different amino acids present in the protein. So, that for that you need to know the sequence of amino acids, but let us say even if I know the sequence, I need to identify whether I am really getting 4 leucines according to the sequence in my spectrum or not. So, that is one task, is to identify the spin system. That is what is TOCSY useful for. But the next task is that which leucine is which number. So, if I have 4 leucines in my spectrum which I am seeing like this, which leucine is which number we do not know. So, that correspondence, that is leucine number, this one leucine is leucine number 4, other leucine is leucine number 20, third leucine is leucine number 16, that kind of a correspondence is not available from this. Okay. So, that part of course comes from another experiment which is 2D NOESY TOCSY, NOESY HSQC, which we will see later, or from 3D NMR experiments. This is what we will see. So, basically one has to build several experiments to get different type of information. That is basically the heart of NMR. In NMR spectroscopy, the whole task is to keep on designing NMR experiments like HSQC, TOCSY, 3D NMR and different experiments, so that we get better and better information about the protein. So, in the next class we will basically look at protein NMR, 3D NMR, and we will see how 3D NMR experiments are built, what kind of information we can get from 3D NMR, and then based on that knowledge we will then start looking at the structures of protein and then start looking at how NMR can be used to solve the structures.